Hi, my name is Kelsey Zagluski, and the question I will be answering is, what occurs when DNA damage detected in G1 is too extensive for repair? So first I will be discussing what is the cell cycle. So here we have a diagram of it. It starts with G1, then goes to S, G2, and M. The G1 phase is growth, S is DNA synthesis, G2 is more cell growth in preparation for mitosis, and M is obviously mitosis, which is cell division. Also, all of the phases in the cell cycle are part of interphase, in which the cell gets nutrients, it grows and divides, but the S phase here, we only have DNA synthesis. So what does G1 actually stand for? G1 stands for gap 1 because it is the first step in the cell cycle, and G2 obviously is gap 2. Here in G1, the cell synthesizes enzymes and nutrients it will need later for DNA replication and cell division. It also produces the most proteins here. This is why the cell grows the most in this phase. And then G1 also has 4 subphases: competence, entry, progression, and assembly. In competence, the cell absorbs nutrients and other elements from outside of the cell membrane. In entry, these nutrients and elements are entered into the cell, and again, they help the cell grow, which is the progression subphase. And then finally, in the last subphase, all of these elements and materials join up to complete the G1 process and end with a restriction point, which is assembly. The next topic I'll be discussing is, how does G1 detect damaged cells? So first, all cells have regulatory aids that monitor growth that happens in the G1 phase. All cells are subject to a restriction point, and this is just to make sure that the cell is on the right track and everything is going smoothly. And this is just a precautionary step that all cells take, and they pretty much just ask the question, is DNA intact for S phase, which follows the G1 phase? And it's just making sure that everything is ready for the next phase of the cell cycle. And these safeguards in the cell that make sure everything is intact are called cyclin-dependent kinase proteins. The next topic I will be discussing is, what are CDKs? So like I just said, they safeguard the process in the G1 phase of the cell cycle, and they also initiate DNA division. Additionally, they engage in biochemical reactions, which regulate steps in the cell cycle, making sure everything is going correctly, and they avoid awry cell division. Additionally, they are enzymes that modify protein substrates by transferring phosphate groups from ATP to stretches of amino acid substrates. And as we can see here, cycline actually allows for the activeness of this enzyme. Without it, there's no enzyme-like activity at all. So as we can see, the cycline is binding to the substrate here, making it cyclin-dependent kinase. Lastly, I will be explaining what happens when DNA damage is too extensive for repair. A process called apoptosis occurs, which is when the cell breaks up its DNA, nucleus, and chromosomes into little fragments called blebs, as we can see here. From there, cells from the immune system come up and consume this debris, or blebs, and which allows for reuse and recycling of these materials throughout the whole cell cycle. On the opposite side of the spectrum, we have necrosis, which is when the cell swells and explodes, releasing its contents, which cause damages to other parts of the cell. This happens when the cell is exposed to chemical toxin or it is mechanically damaged. Apoptosis is a natural programmed cell death, and because it's actually good because too many cells can cause cancer and mutations. Here we just have a quick comparison between necrosis and apoptosis. Here we have cell volume increasing and cell shrinkage.